Brain aging linked to genetic control breakdown. Mouse study reveals aging in the brain isn't simply wear and tear. It's a fundamental loss of control over how genes function, according to a new study using mice. Researchers have mapped epigenetic changes across the brain, revealing a gradual decline in the chemical signals that regulate gene expression. This research provides the most comprehensive epigenetic atlas of aging to date, offering critical insights into why brain function deteriorates with age and potential ways to intervene. The epigenetic landscape of aging our DNA isn't the whole story. Epigenetic markers, tiny chemical tags attached to genes, dictate how those genes are used. These markers change over time, and scientists have used them to create aging clocks that estimate biological age in many tissues. However, the brain, with its long-lived neurons, requires more detailed study to understand aging processes. The new study, published in Cell, analyzed over 200,000 brain cells from mice at different ages, 2, 9, and 18 months. Researchers sliced the brains into ultra-thin sections and examined key epigenetic signals, including DNA methylation and chromatin structure. The results show a clear pattern. As mice age, their genomes lose their ability to precisely control gene expression. Losing control, methylation and jumping genes. One critical change is the loss of methylation, where chemical tags are removed from DNA. Methylation typically silences genes, and its decline in aging mice led to unexpected gene activation. For example, immune genes in brain cells, microglia, became overactive due to a loss of silencing tags. This is concerning because unchecked immune responses can damage delicate brain structures. The problem is amplified by transposons, also known as jumping genes. These repetitive DNA sequences can copy and paste themselves around the genome, disrupting gene expression. The study found that demethylation happens at transposon sites, potentially triggering widespread genetic chaos. According to geneticist David Sinclair, these jumping genes may be a hidden key to brain aging. These are genes we've largely overlooked, yet they track remarkably well with aging, he notes. Chromatin structure and aging signatures. The study also examined chromatin, the protein DNA complex that organizes our genes into chromosomes. Aging brains showed increased topologically associated domains, TADs, tightly packed loops within the genome that compartmentalize gene expression. These extra TADs may serve as a new biomarker for aging, indicating a breakdown in genomic organization. Implications for human brain aging. The loss of genetic control has serious consequences. Overactive jumping genes can trigger immune responses that kill brain cells, disrupting neural circuits. Interestingly, superagers with exceptional memory performance may have lower jumping gene activation, keeping neurons alive longer. This suggests that slowing epigenetic drift could be key to preserving brain function in old age. The research team now aims to apply these methods to the human brain, sequencing epigenetic changes across different ages. The goal is clear: to understand and potentially reverse the mechanisms that drive cognitive decline. Aging isn't just wear and tear, it's a loss of control over how genes are regulated. Joseph Ecker, Salk Institute.